can't grow till December, so that kind of thing.
All right, welcome back. I'm not doing Socrative today. On a review day, I typically just skip the attendance and just give it to everybody. And I've already gotten a ton of emails from people who will be absent today, so I'm just not going to worry about it. We are going to start the lecture with some new content, smooth and cardiac. Again, this is a very basic introduction to these concepts. You will hear more about both of these in A and P2 if you continue on with prepared course. Once we have finished this short lecture, I will then jump over to the review PowerPoint that I put up this morning. We will get through as much as I can. I don't know time-wise exactly how long it will take us. That is a lot of kind of interaction. It's a lot more like those activities that I've been doing. And the one I put up is redacted, so I took a bunch of things out of there, animations and drawings and such. After the class time is over, the full review PowerPoint will post, so anything that I redacted will be on there, all right? And then, obviously, Monday is our next exam, same format as before. I'll be in class if you want to come in and take it. And the weekly quiz should also be up. Hopefully I got all of that done accurately. All right, so we're going to start with smooth muscle. And really, in the context of this unit, what I'm focused on is how do these other muscle categories compare to skeletal muscle, which is the focus of this unit. So we're going to do a lot of comparing and contrasting. Smooth muscle, obviously, does not move the skeleton. It is only skeletal muscle that is attached to the skeleton for the purpose of moving that structure. Instead, smooth muscle is going to be found lining most of our organs. So the entire digestive system, which is one long tube, has a layer of smooth muscle surrounding that entire tube. The heart has cardiac muscle. The uterus is a great example. It's lined with smooth muscle. The bladder has smooth muscle on there. Anything that's going to need to squeeze or stretch in any way is going to have a significant amount of smooth muscle lining it. That smooth muscle is going to be organized in a circular manner. So all of the muscles we saw in skeletal were mostly linear, maybe a little fan-shaped, but they all had a very distinctive muscle fiber direction. These will all be circular, rounding out the sides of whatever organ we're lining, so that when they do contract, because they contract just like a normal skeletal muscle, they're shortening and they're restricting that tube. They're going to, I'm blanking on words today. What opposite of dilate? Constrict. There we go. We are going to constrict whatever structure we are squeezing. Squeezing can do a couple of different things. In the case of the digestive tract, it helps to move things through that long tube system. So one of my favorite examples is the esophagus. The esophagus is a long muscular tube that goes from essentially the base of your oral cavity down to your stomach. Its sole purpose is to move food from having swallowed it down to your stomach. It doesn't do any digestion. It doesn't break down food in any way. Its only purpose is to move that food. And it does so by constricting that smooth muscle behind whatever it is you are swallowing and squeezing it down that tube. Now we think that happens regardless, right? It's just going down. But you can swallow if you're laying flat on the ground. If you are doing a headstand, you can still swallow because it is a muscular process, and that allows that process to happen. It can also help reduce flow of something through an open space. So lumen is just a term in anatomy that means open cavity. So anything that's got a hollow pocket in it has a lumen. So if we are looking at blood vessels, for example, your blood vessels are tubes. They have a hollow space on the inside. They're going to have a very thin layer of smooth muscle on the outside, and they can constrict that and reduce blood flow to different parts of the body for various reasons. The overall structure of a smooth muscle cell is different to a skeletal muscle. So when we looked at skeletal muscle, it was that nice kind of cylindrical structure, elongated. Smooth muscles are going to be tapered. I always think they kind of look like a little slug or a little worm. They are described as having a sigmoidal appearance. Sigmoidal just means S-shaped in structure. And it will not have striations. So remember, that was one of the defining features of skeletal muscle, especially under thin section. It has that distinctive banding that's created by lining up all the sarcomeres. That structure does not exist on our smooth muscle cells. So if we look at a picture here, here's my skeletal muscle. I've got all of my myofibrils lined up, which means all of those sarcomeres are creating those linear I bands and A bands, so you get this kind of banding structure on the surface. Skeletal or smooth muscle, much more wavy in structure. You can see the tapered ends of each of those little cells, and there's no striation to it. There's no lineation created. So the reason smooth muscle doesn't have striations is because they lack myofibrils. They do not have that organelle in skeletal muscle that is distinct for the contraction of that muscle cell. We're going to contract smooth muscle in a different way. So they don't have myofibrils, which means they don't have sarcomeres, but they still have actin and myosin. They're just going to arrange it in a different method. So, lacking striations. That actin and myosin are going to be held in little bundles. Their orientation is still the same, so actin and myosin are sitting one after the other, slight overlap. But we're going to bundle collections of those actin and myosin filaments across the surface of the smooth muscle cell. And they are just randomly scattered. It does not cover the entire surface. There's just pockets of them across the surface of that cell. That means overall, compared to skeletal muscle, there are far fewer actin and myosin. So the overall contraction is going to be reduced because we don't have as many of those fibers that are going to be overlapping during that process. So they are going to do less shortening, and in fact, the overall result of the contraction of a smooth muscle cell looks much more like bulging. So it's going to shorten, but it's also going to kind of spread out to either side. Those actin and myosin bundles are going to be attached to this very net-like system, it's a fibrous network, called intermediate filaments. So there are going to be these little anchor points across the surface of my cell, and then there will be little fibers connecting each of those anchor points. We then put our actin and myosin bundles anchored to that net, and when the actin and myosin overlap, what they're doing is pulling on the net. So they're not physically constricting or shortening the muscle cell. What they're doing is squeezing it, and they are bulging it out between that fibrous network. So I always like to picture it, if you've ever played with one of these little stress balls, you squeeze them and it bulges out in between that little net that's holding it all together. That's the same kind of concept of how smooth muscle is contracting. It's bulging within this net that's being pulled on. So the sigmoidal shape, that S structure, the lack of those distinct myofibrils and instead having that random assortment of actin and myosin bundles means that our smooth muscle can really stretch. So it does contract, it absolutely constricts, but its best quality is the fact that it can stretch to accommodate things. So when we are swallowing, again, we're constricting behind it to push that food down, but the food itself is expanding that tube. It needs to stretch to accommodate that while you're swallowing. Same thing with your bladder, for example. As the bladder fills with urine, it needs to stretch to accommodate that. And it's these smooth muscle cells and the way they're designed that accommodates that stretching. Before I move on, I wanted to jump over to biodigital real quick. So there is an animation. I don't know if it's saved into the file or not, but this is a smooth muscle, so you can see that network of fibers, our little anchor points, and our actin and myosin bundles attached to that fibrous net. And if I go to the animation slide, so the actin and myosin are contracting, they're pulling on that net, and it causes it to bulge out to either side, but then it can also stretch because it's not restricted by those myofibrils. So I think it's a good visual. Okay, back to my PowerPoint. All right, activation of skeletal muscle. We talked extensively about this in the physiology lectures. It's only going to be activated by those neurons. So those neurons are coming down. We go through that whole action potential, release of acetylcholine. That's going to start an action potential in our muscle cells. Smooth muscles can also be activated by neurons. There are nerves that go down to the smooth muscle cells. But hormones can also trigger a contraction, particularly in the digestive tract. The digestive tract has its own monitoring system that you are completely subconsciously aware of. You have no notion of when food is moving from one place to the next. It's completely controlled subconsciously. And a lot of that motion is being controlled by the presence of different chemicals. So that's another major difference between skeletal and smooth. Skeletal is completely consciously controlled. You get to decide when those muscles contract. How do they contract? Are they contracting quickly, smoothly, gently, short? All of those things you get to decide. We do not control smooth muscle. You don't get to decide when your blood vessels are constricting. You don't get to decide when your stomach constricts and puts things into your duodenum. All of that is completely subconscious. You are not aware of it. So both the hormonal and the neurological control for your smooth muscle is all subconscious. Now, just like skeletal muscle, we are relying on the presence of calcium for that purpose of contraction. Remember, we had the action potential come down our T-tubule. That was going to trigger the release of calcium from my sarcoplasmic reticulum. Calcium bound to my actin. Actin rotated, exposed my active site, and then that allowed myosin to bind, complete my power stroke. In the case of smooth muscle, the calcium doesn't bind to the actin, so there's a difference here. Instead, the active sites are already exposed in smooth muscle. What we need is the calcium to bind to the myosin head, and that gives it the power to bind to the actin. So it's just a slightly different method of achieving the same process. Once that myosin head has bound to the active site, moves in a power stroke, ATP is still used to reset the process. So actin does not need calcium to rotate. It's already rotated. Calcium is going to bind the myosin head to the actin itself. And here's another figure showing that. So we've got calcium that's going to be released. That calcium is going to bind to the head of my myosin, allow that to bind to my actin, and result in a power stroke. Smooth muscle also has gap junctions. Is that something you guys covered earlier on in this class at all? Well, wasn't sure, so I figured I'd still cover it. So gap junctions are a connection between adjacent cells. It is a physical tunnel between the plasma membranes. And what that does is connect the cytoplasm from one cell to the cytoplasm of the next. If I generate an action potential on my smooth muscle, I've triggered the process of contraction. That can propagate down my plasma membrane like normal, but now rather than just staying on this one cell, it can propagate directly onto the one next to it because they're already connected. So what this does is allow the process of contraction to spread smoothly across all of the connected smooth muscle cells at once. Skeletal muscle does not have gap junctions. We don't want it to have gap junctions. We talked about motor units and how we can recruit different numbers of them to achieve very specific motions. If they were all connected via gap junctions, you lose that specificity. You can no longer precisely pick something up. You can't do it gently or strongly. You lose that ability to control how your muscles are moving. So gap junctions are great for smooth muscle because we want all of the gut tube to contract as one, but we don't want our skeletal muscle to operate that way. So as an example, the smooth muscle with the digestive system, we want that contraction to happen completely across the entire tube and to move continuously as it's squeezing food through the small intestine, for example. We want that to be one fluid motion. So that synchronicity that's achieved through gap junctions is beneficial for the purpose of smooth muscle. Where do we find smooth muscle? Mostly in the digestive tract. Your entire system, from the esophagus to the stomach to the small intestine, large intestine, all of that is going to be lined with smooth muscle. Lining specific organs, the heart is the exception because that's going to be cardiac muscle specifically, but things like the bladder and the uterus. Lining blood vessels, all of your arteries, all of your veins have a very thin layer of smooth muscle that allows them to constrict and redirect blood flow. And associated with the eye and your hair follicles. So if you've ever experienced goosebumps, where your hair stands on end on your arm, there are teeny tiny little smooth muscle bundles anchoring the base of every single one of your hair follicles. And that smooth muscle constricts and pulls that hair up. That is the physical response of goosebumps. And that's a subconscious thing, right? You don't get to decide when that happens. So that's an indication that you're looking at smooth muscle function. Same thing with the constricting of your pupil. That's going to dilate and constrict according to light coming in. You don't get to decide when that happens. It's an automatic response. Smooth muscle is controlling that. Oop. So physically, it's good to be able to distinguish smooth muscle from skeletal in tissue slides. It's going to have a much more wavy texture to it, much more irregular. I always note that the nucleus or nuclei tend to look very elongated or stretched in a smooth muscle section. Again, that lack of striations, and there's no distinct orderly arrangement to it. I stress this because when you go into A&P1 or A&P2, we just did a lab last week. They had 5 different tissue slides to look at, and all of it was, where's the smooth muscle? How thick is the smooth muscle? Which way is it oriented? So being able to recognize it is going to be helpful to you in the future. All right, cardiac muscle. This is going to be much shorter because I don't want to say a whole lot. There's an entire lecture dedicated to this in A&P2. It is striated, so we are going to still see that lineation of myofibrils, but it is specifically striated muscle of the heart. Cardiac always refers to heart in some function. The cells of the heart are called cardiomyocytes. So because they are specific, we've given them their own special name. Rather than just smooth muscle cell or skeletal muscle cell, these are going to be cardiomyocytes. So found in the walls of the heart, its job is to contract and compress the walls of the heart, thereby pushing blood through our circulatory system. It is going to have sarcomeres bundled into your myofibrils. We're going to line them up, and so you're still going to get a striated appearance. But because of the interfingered nature of the cells of cardiac muscle, it's not always going to be a perfect line. So you'll see striation, and then it'll jump over a little bit, and you'll see striation over here, and then it'll jump over here. The interfingered nature that I'm describing is this splitting of cardiac cells. So see how this one cell comes this way, and then its fibers are suddenly running over in this direction, and they keep going in this direction. They split and they meet up with each other and they bump into each end and they run back and forth in these zigzag patterns. All of this is making sure that they have really good communication between the cells, because more than anything, you want your heart to contract as one. You don't want that muscle to be like, you need that to contract as one single contraction so that it is pumping blood efficiently through your system. You connect those interfingered cells with these little structures right here. These are called intercalated discs. They are often seen in tissue slide as little purple or pink lines. And that's going to be a point of communication between adjacent cells. Those discs consist of interfingered membranes. So the actual plasma membrane of those cardiac cells fit together like this. Lots of surface area for lots of communication. Desmosomes, those you can think of as like little pieces of Velcro that hold those cells next to each other together so they don't pop apart. And just like in the smooth muscle cells, we have gap junctions so that they can communicate those action potentials so that they can contract as unison. So the overall structure of cardiac, the biggest thing is that we want it to contract as one unit. The contraction impulse for cardiac muscle is different. So we have neurons going down to our skeletal muscle, again, that whole acetylcholine process. The cardiac muscle has its own internal system. It's called a pacemaker. You may have heard of a pacemaker in medical terms. The artificial pacemaker is replacing the natural function of the the natural pacemaker that we are all born with. That pacemaker is generating spontaneous action potentials all day long, every day, creating your heart contraction. All right. And then the last bit here, the last major difference of cardiac muscle to smooth or comparing it to skeletal muscle is that in our action potential, we go up, we've depolarized, rather than immediately dropping back down like we saw in skeletal, there's a plateau phase. This is caused by an influx of positive ions. I'm not giving you the details here, you don't need to worry about the detail. But a bunch of positive ions are flowing into the cell. That's prolonging the positive stretch before it repolarizes. And again, what this is doing is allowing all of those cells to receive the signal, and then we're going to contract. It's going to make sure that that signal has been received by everything before you start the contraction process, because it is so important that your heart muscle contracts as one. All right, that's all I had for you for that one. Does anyone have any questions? I did go through very fast, but okay. Then I am going to jump over to my other PowerPoint. So the way I typically do reviews is, again, we'll do a lot of activities. Some things I'll just mention. I tend to pick things that I consider to be key topics from that unit, and since this is our first unit where the whole thing has been presented by me, I feel like we can cover it pretty effectively. The other thing is at the end, if I don't get to it before we have to leave, there is a slide that lists, I like to summarize potential short answer questions. So I'll just list anything that I could consider to be a good short answer style question. So when you're practicing this weekend, you can kind of focus in on that for any kind of written response. All right, so this is not an activity. I'm just reiterating those muscle charts. They're still important. So be able to identify any of these muscles in a figure. I would have an arrow pointing to them in some way. Describe the groupings, so in particular the names that I applied to things, so like quadriceps, which muscles belong to quadriceps, or where do you find quadriceps in the body, be able to describe that position. Know things like superficial versus deep, so which of the muscles are superficial on the forearm, which of them are deep. Can you describe that kind of concept to me? And then being able to interpret motion in particular. So if I had a picture of an arm with an elbow here, and I had an arrow pointing it that way. This motion, what kind of motion is that at the elbow? Be able to describe those sorts of things. So that's what I really want you to focus on for the muscle charts. There will be some amount of ID. The ID is much more focused in the lab, so I'm not going to flood the exam with ID questions. So muscle groups, I already talked about this, but all of the groups that I mentioned, I had a slide summarizing all of the ones that I wanted you to know. Make sure that you can describe where they are, which muscles make up each of those, what kind of major motion at what joint each of those groups are responsible for. So there's an example. I already used the exact same example I was going to talk about. All right. So if we want to use this as an example, the quadricep muscles, those are my 3 vastus plus rectus femoris. What joint do they move? What joint are these moving? Based on this picture. The knee, right? Why are they not moving the hip?
They don't cross over the joint?
They don't cross that joint. These muscles all originate on the femur. If I want it to move my hip, they need to start up above that hip joint. So you can interpret the motion just by looking at the attachment points. Again, you don't need to know the specifics. You don't need to know that vastus lateralis attaches on the greater trochanter. You just need to know that it's on the femur. And then what joint, they do move the joint, or they move the knee. What motion are they going to generate?
Extension.
They're going to generate extension, right? Because extension for the knee is moving forward, expanding the angle of that joint. All right, we've seen this before. Muscle organization. Take a few minutes, talk to your neighbors. I want to hear chatter. See if you can put these things in order from largest to smallest. In addition, there are terms in there that apply to the protective layers that surround our different levels of muscle, as well as some categories like cell and organelle. All right, it's getting quiet, so we'll run through it. What is the largest level of muscle organization, the largest structure that we provide a name to? The muscle body, yep, so muscle body's first. What do we bundle together to make a muscle body? The fascicles, yep. Whoop, well, there's the next answer. We bundle together fibers to make fascicles. What do we bundle together to make muscle fibers? What special structure is in them? Myofibrils. All right. What are myofibrils chock full of? Sarcomeres. And each of those are going to be bundling what? Actin and myosin. Excellent. All right. So that is the muscle organization of structure from largest to smallest. Remember, actin and myosin is the contracting point. As those contract, that shortens our sarcomeres. That shortens the myofibrils within our muscle fibers. All of our muscle fibers contracting as one are contracting the muscle fascicles, which result in the shortening of the muscle body. All right, what is the protective coating that we surround our muscle bodies with? Epimysium, good. Muscle fascicles are surrounded by what? Perimysium. And then muscle fibers would be surrounded by the endomysium. Those are your three mysiums, three sheet layers. And then which of these structures represents the cell in our muscles? Muscle fiber, and I scrolled too far again. Muscle fiber is the cell, the myofibril is the contracting organelle. All right? Any questions on that one? All right. What are the 5 muscle arrangement categories that we talked about in class? So there were different shapes of muscles that attributed their type of contraction, how strong they could contract, how completely they could contract. Take a minute, see if you can talk to your neighbors, see if you can come up with any of them. All right, can anyone give me a name of one of those fascicle arrangements? Pennate? Yep, pennate's one of them.
Parallel.
Parallel, we've got another category. 3 more. convergent, and circular, yes. That is exactly the order I listed of them. I'm assuming you're looking at your notes. That's perfectly fine. So for each of these, what is the overall structure? How would you describe a pennate muscle? Like a feather. Pennate means feather. So you're going to have a long tendon running down the center of that muscle body, and the muscle fibers are going to spread out to either direction from that core tendon. That's your pennate organization. What about parallel muscles?
Unparalleled fibers.
All the fibers are running in the same direction. They're all going to run parallel to each other. What about fusiform? If I told you an example of the fusiform would be like the bicep muscle.
Just like converges on both ends.
Converging on both sides. So you've got a tendon on one end, a tendon on the other, and your muscle fibers are going to run from one tendon to the next, constricting to one end on each side. Convergent muscles then, how are those different?
Only on one side. Like
Only on one side. So this time I've got a tendon on one end, and then all of my muscle fibers fan out and converge to that single structure. And then circular muscles are a little bit more intuitive. Those muscle cells are running in a circular manner. Things like the surrounding of the eye, when it constricts, it squeezes the space around your orbit. So being able to describe the structure of each of these, and I also recommend being able to provide a muscle example. So for example, the pennate, this kind of thing, one of the classic examples would be, now my mouse isn't working, the deltoid muscle. Deltoid sits across your shoulder, spread out along here, one single tendon that runs down the outside of the shoulder, and all of those fibers converge towards it. Parallel muscles would be any of your abdominal muscles. The rectus abdominis is a great example. Things like sartorius or gracilis. Those are also long, skinny muscles, but all of their fibers are running in the same direction. Fusiform, your classic muscle shapes of biceps, triceps, any of the muscles that we were looking at in the arm, those are all going to have that same structure. Convergent muscles, pectoralis major, broad connection all across the sternum and the clavicle, all of those fibers converging to a single point on the underside of the arm. And then I already mentioned the eye as an example of your circular muscles. Right? So the different ways in which we arrange these. Which one of these categories has the most range of motion? The most length along which they can contract? Not the fusiform. The parallel ones, yep. So the parallel have one long length of contraction. There's no convergence at any point that's going to restrict that contraction. The entire length of that muscle can contract as one. So the greatest range of motion of contraction belongs to our parallel muscles. Which one of these categories, technically two of them work, gives us the strongest types of contraction? Convergent and pennate muscles, yep. All right, action potential. Could you draw me a graph? Could you set it up? So talk to your neighbor, see if you could build an action potential graph.
No, yeah, I'm sure, yeah. I was making sure of that one. I have no idea.
All right, so what belongs on my x-axis? What label would I put down here? Time, yep. Time is increasing in that direction. What label goes on this side? Voltage, yep. I can write sideways. And voltage is increasing in that direction. Where do I put my 0? This is the tricky one. Do I put it here? No. All right, we can all agree that 0 is going to go about halfway up the graph. So then it's negative down here and positive up here. Remember, I'm not particularly caring whether or not you know the numbers. It's more about the change in polarity, so going from negative to positive. So our chart is going to start at rest, in the negative. That's where our cell is at rest. What needs to happen to start this process? We need to get a stimulus. So stimulus is received, and that's going to start moving ions such that we head up in polarity. We're going to go towards the positive. What do we have to pass to make sure we complete this structure or this process? The threshold. That threshold, sorry, chalk is not something I'm used to writing with. Threshold is below 0. We pass that threshold. We initiate the rest of our action potential. What is the normal pathway from here? Do I plateau or do I go straight down? Straight down. When would I plateau? Cardiac muscle. We just talked about that one. So that's the difference there. All right, what happens at this point? We hypercorrect. We go past the resting point, and then we're going to reset back to our normal pace. So very basic understanding of that graph. Now, can you give me more specifics? Talk to your neighbor. Could you tell me what's happening at each one of those dots? All right, give you another 30 seconds and we'll run through it. Okay. So my first dot, right there at the stimulus. First of all, what is the stimulus? This isn't going to be on this PowerPoint, but what is the stimulus?
Calcium goes and attaches to the sodium channel.
Yes, in a more general sense, though. Where is it coming from? Is it from a neuron? Yep. And that neuron is going to be releasing acetylcholine, a chemical signal, across, what was that gap?
Synaptic cleft.
Synaptic cleft or neuromuscular junction. Both are acceptable for that. All right, so we've released a signal from my neuron. Where is my mouse? There we go. That is starting a stimulus, starting an action potential. What is happening now on this graph? Something's opened. Ligand-gated sodium channels. Again, that ligand means chemical, so the chemical is the acetylcholine coming through that neuromuscular junction. All right, my next dot at my threshold. So my voltage is changing, I'm going more towards the positive. My voltage has changed such that, what happens next? Voltage-gated what channels? sodium channels are going to open. So now I've got a whole bunch of sodium flooding into my cell. All of these positively charged ions are coming in, and that's going to shoot straight up the side of my graph. So what word would we use here? Depolarization. Yep, depolarization goes there. And the description would be that my polarity is flipping. At this point, I have gone from negative to the positive. All right, next dot. There are two things that are going to happen here. The sodium channels are closing. Yep, no more sodium movement. What else? Potassium are what? Opening. Are they voltage or ligand gated channels? Voltage gated channels. So my voltage gated, technically all of my sodium channels close, and my voltage gated potassium channels are going to open. What happens along this portion of the graph? Repolarization. So depolarization is going up, repolarization is going down. That means my polarity has flipped back to the negative. Down here, what is happening / what is the name for this? Hyperpolarization. And what are we changing? Always look at the process and think, what is still open? My potassium channels need to be closed. So my voltage has changed such that that channel can no longer remain open. I have dropped so far in the negative, I'm out of its range of operation. Those channels close. So now nothing's moving, right? I've closed all of my openings, we've restricted that motion. The last bit here, what do we need to do? Active transport of both potassium and sodium back to their original concentrations. There's your action potential. Okay. So be able to describe this graph in a few sentences, or if I gave you a couple of points on there, be able to tell me exactly what's happening at each of those points and how that's going to be tied to the process. All right, and then depolarization, repolarization, hyperpolarization. I had them on there just later. So we generated an action potential in my muscle cell. That action potential is propagating down the sarcolemma, my plasma membrane, and it's going to dive down what structure? The T-tubules. The T-tubules are in a triad, a set of 3 tubes. T-tubule in the center, it's sandwiched between 2 tubes that are making up part of the sarcoplasmic reticulum. So what am I opening within this junction between these structures? What voltage-gated channel? Calcium. So it is the action potential, the change in voltage, passing over that connection with my sarcoplasmic reticulum that causes that voltage-gated channel to pop open. So calcium is now flooding out of my sarcoplasmic reticulum. Where is it flooding into? What region of the cell? Sarcoplasm, which I would accept cytoplasm. They are equivalent. Sarcoplasm is just the term we use specifically for muscle cells because anatomists are weird that way. All right, so I've flooded that space with calcium. And remember, my sarcoplasm is the space between the plasma membrane and all the things inside the cell. We've stuffed our cell full of myofibrils, my contracting unit. Myofibrils are full of actin and myosin. So calcium is going to bind to that actin, and what's it going to do? Rotate it. The myosin is now able to reach a specific structure on the actin. What is that structure? The active site. The myosin head binds to that active site. It's going to pull on the actin. What is that motion called? The power stroke. And then what do we have to spend to reset that process? ATP. Remember, every contraction of a muscle is an energetically expensive process. We have to spend energy to do active transport at the end of every action potential, and every contraction is made up of multiple action potentials. We have to spend energy in order to bind the myosin head. We have to spend energy to release the myosin head. All of those processes are going to require this the spending of energy. And then at the very end, we have to spend energy to put our calcium back into our sarcoplasmic reticulum. All right, we've got 4 more minutes. I'm not sure we'll finish, but we'll see what we can get through here. Talk to your neighbor. See if you can come up with 4 different things that are going to determine the strength of a muscle contraction. What sorts of things can we do to improve the strength of that process? All right, so can anyone give me something that we can do to increase the force of a muscle contraction?
Number of motor units recruited.
Yep, how many motor units am I going to recruit? What is the definition of a motor unit?
So you have to group that the neuron is signaling.
So every muscle neuron, I've lost my chalk, hold on. Yep. Neurons coming down. In the case of skeletal muscle cells, it's going to split into several little fibers, and each of those are innervating a different skeletal muscle cell. So my motor unit is what is the grouping of muscle cells that are innervated per single neuron. So you can change the strength by changing the number of motor units you recruit. If you recruit more motor units, you can have a stronger contraction. If you only send a signal down a handful of motor units, you'll have a slower or a more gentle or a more precise muscle contraction. So, motor units. What else can we do? The size of my muscle cells. So I can increase the diameter of the muscle fibers. By increasing the diameter of the muscle fiber, so again, fiber is equivalent to cell, by going from this to this, I've increased the number of myofibrils inside. I've just made more room. So I've got 4 in this one. I've got room for 8 in this one. The more myofibrils, the more actin and myosin you have, the more structure is contracting as one. What you don't do is increase the number of muscle cells. So when you are bulking up muscle to increase strength, you are not increasing the number of cells, you're just increasing their size. I had a picture for that too. What else can we do? What sends the signal for muscle contraction? What's the process we just practiced? An action potential. So if I wanted to have a really strong, intense contraction, what would I want to do with those action potentials? Send more of them. The frequency of that signal. You only send a couple, and you're going to have a short contraction. It'll be a pretty weak contraction. Remember, we never send one. That is a completely theoretical concept. But if you want to have a really strong contraction, you're going to send repeated action potentials one after the other. And you'll keep sending those signals until you're done with that contraction. Last thing that we can do, am I going to have a stronger contraction? Sorry, it's the end of the class, so I'll finish this. Stronger contraction when I'm starting like this, or like this? Somewhere in the middle, where my actin and myosin are mildly overlapped. There's still room for contraction, but they have a good enough starting point. So the length of muscle fiber contraction. So I'm just going to scroll through these, comparing fast and slow twitch muscles. Make sure you can go through that. How can you compare the different types of cells? So skeletal, smooth, and cardiac. This PowerPoint listed all the things that we just went through side by side. So feel free to look at that. The whole thing should be posted within the next 30 seconds, so you'll have access to that on Blackboard. That is all I have for you, so have a good afternoon and weekend.